Hello everyone, this is R. Rajarajeshwari working as an assistant professor in the Department of Biotechnology, Kamaraj College of Engineering and Technology. In this video, we are going to talk about the topic biological methods of nanomaterial synthesis. Usually the synthesis of nanomaterials falls under three broad categories: physical method, chemical method and biological method. In physical method we have methods like ultrasonication, physical vapor deposition, etc. Whereas in chemical methods we have methods like chemical vapor deposition, colloidal synthesis and self assembly. Whereas in biological methods there are many types of methods are available, from which 4 such methods will be focused in this lecture. So firstly we are going to see about plant mediated synthesis for nanomaterials. So the plant mediated synthesis is a very eco-friendly and cost effective method that can be used that is widely used in the nanomaterial synthesis. So basically this method falls under the category of green chemistry, where more greener approaches are used and environmental pollution is controlled to an effective extent. So these are the steps that are coming under the plant mediated synthesis. So firstly, extraction of plant biomolecules from the plant extract, and the next is reduction of metal ions, and the next step is formation and stabilization of the nanoparticles, and the final step will be characterization and post synthesis treatment. So firstly, plant extract is prepared. So the type of plant and the material that we are using is to our choice. So basically we are going to choose the plant material and we are going to prepare an extract from it, and this extract is introduced into the metal ionic solution. Say for example, if you are going to prepare gold nanoparticles, this plant extract can be introduced into a solution called gold chloride solution, in which biological reduction reaction happens. Under the biological reduction reaction, first step will be nucleation, and the second step will be accumulation, and the third step will be formation of stabilized and capped nanoparticles. So here the 4 important features has to be focused while carrying out this method. They are pH, concentration, reaction time and reaction temperature. So these 4 factors will play an important role in the formation of the nanoparticles, and they will determine the quality of nanoparticles that are going to be formed. So this method is a very widely used method, and this is the first basic method under the category of biological methods. So next method is microbe mediated synthesis for nanomaterial synthesis. So the microbe mediated synthesis of nanomaterial involves the use of microorganisms like bacteria, fungi, algae, etc. for the formation of nanoparticles. So in this microbe-mediated synthesis, 2 main categories are seen. The first one will be extracellular synthesis of nanomaterial, and the next will be intracellular synthesis of nanomaterial. So in this extracellular synthesis, we can see that the microbes are introduced into a medium containing the metals, where reduction reaction happens. The enzymes and proteins that are released from the microbes will reduce the metals into nanoparticles. So basically the reaction happens outside the cell, and the factors that are going to help in the formation of nanoparticles will be released from the cell. So in the intracellular method, whatever metals we are going to reduce is introduced inside the cells, where the components of cell, the materials that are present inside the cell is going to reduce the nanoparticles, and after which the required material can be extracted from the cell. So here these are the steps that are involved in the microbe mediated synthesis. The first will be selection of microorganisms, and the next step is culturing the microorganisms, and the next step will be preparation of the metal ion solution. So the next step will be exposing the metal ion solution into the microbial environment, and next is formation and characterization of nanoparticles, and the final step will be post synthesis treatment. So here if we can see, the metal ion is first introduced into the microbial environment. In case of extracellular synthesis, the microbes and the metal ion containing medium is introduced together, whereas in intracellular method the metals that has to be reduced is introduced inside the cell, and after that reduction reaction happens, where metal ions undergo nucleation, accumulation and capping process happens, after which the nanoparticle can be obtained. So the next method will be enzyme mediated nanomaterial synthesis. So, in this method a specific enzyme is used for the preparation of nanoparticle. Depending upon the metals that we are using and depending upon the required component that we have to obtain, the specific enzyme can be shortlisted and we can prepare it or we can purchase it. So, in this method, so basically enzymes are biological catalysts that accelerate the chemical reaction without taking part there. So, here the enzyme assist in controlling the size, shape and morphology of the nanoparticles that are being prepared. So, this is a very eco-friendly process again. So, these are the steps that are involved in enzyme mediated synthesis. Here if we can see, the first step will be selection of enzyme and the selection of metal source and the enzyme preparation. The next step will be interaction of enzymes with the metal ion solution, and the next step will be formation of the nanoparticles, and after that characterization and purification post treatment can be carried out. So, here in this image if we can see, gold salt is being introduced into an environment where 3 different enzymes are used, and based upon the enzymes we are getting 3 different products, that is 3 different nanoparticles with 3 different categories. From this we can conclude that depending upon the enzyme that we are using, the resultant nanoparticle characteristics might change. So, the next method will be exopolysaccharide mediated nanomaterial synthesis. So, exopolysaccharides are high molecular weight carbohydrates that are being released by the microbes into the environment, that acts as a protective covering for the environment. So, the next method will be exopolysaccharide mediated nanomaterial synthesis. So, exopolysaccharides are basically the high molecular weight carbohydrates that are released by the microbes into the environment, that acts as a protective layer for the microbes. So, these exopolysaccharides are isolated and they can be used for the synthesis of nanomaterials. So, here if we can see, exopolysaccharide is introduced into a medium containing Ag metals, where these exopolysaccharides are getting attached to the surface of the metals and the reduction reaction happens. Once the reduction reaction is complete, capping will happen. Here in this process, instead of introducing a separate capping agent, here what happens is the exopolysaccharide itself acts as a capping agent for the nanoparticle. So, the nanoparticle becomes stabilized and agglomeration is avoided, and once the nanomaterials are synthesized, various characterization techniques can be employed like SEM, TEM and AFM and XRD methods can be employed. After characterization, post treatment and purification steps can be carried out. So, these are the some of the biological methods under the nanomaterial synthesis. Next is chemical mediated nanomaterial synthesis. Here we are going to see about chemical vapor deposition. So, chemical vapor deposition is a widely used chemical method for the synthesis of nanomaterials and thin films. In this process, gaseous reactants or precursors is introduced into a vacuum chamber where they react with the substrate and they form a thin layer of nanomaterial, and the byproducts are removed from the gas outlet. So, this is the overview of the process. So, here the process is carried out in a vacuum chamber, and again upon heating of the precursor, the gaseous precursors are introduced into the gas inlet where substrate and precursor reaction happens. So, the key process involves precursor injection, chemical reaction and nanomaterial formation. So, this is the setup of the chemical vapor deposition. So, this is the gas inlet and this will be the gas outlet. So, RF coils are wound around the chamber, and the whole chamber is will be kept in a vacuum condition. So, first step will be introduction of the precursors. So, precursors are heated up to more higher temperatures of around 3000 to 4000 degrees Celsius, and the precursors are introduced into the chamber. This will be the first step. Once the precursors are introduced, they are going to react with the substrate. So, substrate and precursor react, and the precursors that are introduced will form a thin film over the substrate. So, the substrate is kept on a thermal insulation to overcome the effects of higher heat of precursors. So, next step will be the byproducts that are removed from the precursors. Precursors will be deposited onto the substrate. So, the byproducts that are remaining will be letting out through the gas outlet. So, this will be the whole process setup. So, for this process, some key parameters has to be noted while carrying out the process. So, the parameters are temperature, pressure, flow rate of precursors and substrate. So firstly, temperature. So the temperature plays a crucial role in the decomposition rate of the precursor and also the crystallinity of the nanomaterial that is going to formed. So the substrate temperature will be around usually 500 to 1000 degrees Celsius, and again the higher temperature is said to improve the crystalline quality, but depending upon the material that we are using, the crystalline quality might change. So the next will be next parameter will be pressure. So the process is carried out at an atmospheric pressure or low pressure. Depending upon the conditions, the CVD can be categorized into two wide categories like atmospheric pressure MOCVD or low pressure MOCVD. Here the word MO denotes metal organic, where metal organic precursors are used for the process. So low pressure will allow for better control of film composition and film thickness, where the atmospheric pressure method is going to help us in the formation of nanomaterials for large scale production. So then next parameter will be flow rate of the precursor. So this is a very important parameter where the precursor and reactive gases is going to influence the growth rate of the nanomaterial film that is going to form on the substrate. So the precise control over the precursor flow is very essential for obtaining high quality of nanomaterial with uniform composition and uniform thickness. The uniform thickness is a very important factor for the use of nanomaterial in wide applications. So the final parameter will be the substrate, where the substrate material can vary depending upon the intended application. Common substrate materials include silicon, gallium arsenide and sapphire, or silicon carbide, etc. So the choice of substrate will influence the crystallinity and morphology of the film that is going to form. So these parameters must be followed, and these parameters must be noted before starting the CVD process. So thank you.